Okay, thank you. So yeah, I'm Mikaël from the University of Lille, and this work was a joint work with people in my group, Camille Marché, as well as master students from the university, and people in Paris working on RNA folding. So first, we were interested in SARS-CoV-2. So you know, I guess, this virus. An RNA virus whose genome is about 30,000 bases per long. And we were interested in the structure of the RNA genome. So the genome folds with stem loops, as you can see on the picture. How can I have the mouse? I don't know. Here it is. Okay, so you can see stem loops here. And we were interested to detect somehow those stems just using genomic data, so genome sequences. So this is a stem loop seen closer. And what we were interested in is what happens when there is a mutation. So for instance, if you have a mutation right here, you have a G instead of the original U. So this breaks the binding between the U and the A. The two nucleotides are not bound anymore. And if this stem is important and matters for the genome, there are good chances that this genome would be negatively selected. On the contrary, if you have an additional mutation, so a C instead of the A, this will compensate for the original mutation, so it is a compensatory mutation, and this kind of double mutations could be positively selected. And that's what we are looking for in the genome sequences. So imagine you have many genome sequences of SARS-CoV-2, and we do have. You have a multiple sequence alignment of those genomes, and you will have some columns here where you have the original U or the mutated G, and in another column far apart, you will have the original A and the mutated C. And so we want to identify such covariations within multiple sequence alignments here of SARS-CoV-2, but it can be any other genomes. So I won't enter into the details of computing covariations and covariation scores, because it's not the focus of my talk, but just to mention a few words. What we were interested in basically is a number of compatible pairs between two columns. Okay, so on the left-hand side, you have two columns where all the pairs are compatible, meaning that they can bind together. While on the right, you have just two compatible pairs. And basically, that's what we have to count in order to compute conservation scores. The drawback is that you have to consider all possible columns in the multiple sequence alignment. So you have a quadratic number of pairs of columns to consider, which is quite large. Knowing that n, in our case, is about 30,000, the size of the genome. And S, it's the length of one column, so it depends on the number of sequences we have. And actually, we have about 4.5 million genomes available in the Hunt et al. Datasets released in 2024. So that's quite a lot of data, and in order to process that, we need an efficient representation of multiple sequence alignments, and so that's the topic of my talk. So we have a multiple sequence alignments like this one, and what we want to do is, well, to gain benefits from the redundancy in the data. So I can show you this multiple sequence alignment in another way. I just highlight the letters that differ from the previous one in a given column. Okay, so for instance, the G, which is here, is different from the previous one in the column, so it is in black, and so on. And we can represent this in a binary way, where black letters are ones and gray letters are zeros. And we need to store on top of that the nucleotides that correspond to each one. And so with this information at the bottom of the slide is enough to recover the initial multiple sequence alignment. Okay, so we have the same information, which is represented here. It's just in another way, and which is way more compressible in that way than in the previous way, because we have plenty of zeros, and so we can compress that efficiently. I will introduce a notation which is I, the number of runs in a column of a multiple sequence alignment. Otherwise stated, it's the number of ones in a column here of my bit vectors. And so if I have a fewer number of runs, I will be able to compress that more efficiently because I will have more zeros to compress, and I will have less nucleotides to store because I have a nucleotide stored for each one in my bit vectors. And we want to query that multiple sequence alignment. We don't just want to store it. So on top of those bit vectors, we have the classical rank and select operations. Rank computes the number of ones in any prefix of the bit vector, while select gives us a position of 1 in the bit vector. So for instance, on that example, a rank at that position is 2 because I have 2 ones in the prefix of the bit vector until that position, while select 2 gives me this position 4, because the second one is at position 4. So the good news is that we are able to use compressed bit vectors and have those operations in constant time, which is nice. And so we can query our multiple sequence alignment. So we can access any nucleotides. So we have random access to our multiple sequence alignment. I won't enter into the detail of how we compute it, but it's fairly easy. I'm more interested in counting nucleotides in columns, actually. So we want to be able to count nucleotides. So what are the nucleotides I have in a column, and how many of them do I have? And to do that, we only need to iterate on once in the bit vector, okay? So if I take this example, for instance, here's the first one. It corresponds to a run in my column of length 1. I have just one nucleotide, 1G. So I have 1G in my column, and I move to the next one, to the next run, which is of length 2. So you have 2 Cs, and then 1 additional G, and then 2 additional Cs. And by iterating on the ones of my bit vector, I'm able to count all the nucleotides in the column of the multiple sequence alignments. And so the time complexity does not depend directly on the number of bits of my column, but only on the number of ones, so the RJs and number of runs in my column. And if I want to count nucleotides in pairs of columns, and that's actually what I'm really interested in, it's exactly the same idea. Conceptually speaking, you can just do a bitwise OR between the 2 columns and proceed as I've shown for a single column. And so in that case, the time complexity also depends on the number of runs in the column. And so we have a time complexity which depends on the number of runs. So if I have fewer number of runs, I will have queries that will be faster. So we have a quite nice property where if we have a smaller number of runs, we will have an index that is both more space efficient and more time efficient, okay? Usually we have a time-space trade-off, and it's not the case here. So if we are able to reorder our original multiple sequence alignment in order to have a smaller number of runs, it will help us to have a more efficient index. So if I take back my example and I reorder some of the sequences, I will have something like that. So you see that there are a smaller number of differences. Okay, I just have here one difference here compared to 4 originally, and so on. So I have a smaller index. But okay, I've done this by hand. But how can I do it more generally? Well, you have a bad news now. This is NP complete to find the optimal order. So we won't try to find an optimal order in that case. Instead, we can rely on heuristics to reorder sequences. One of the heuristics is to use the phylogeny. So that's inspired by what have done Brinda et al. In the 2025 paper on compressing using the phylogeny, compressing genome sequences using the phylogeny. So that's one possibility, but we have to know the phylogeny, or we have to compute it, and it can be quite expensive. So we wanted also to introduce a reordering that would only depend on the multiple sequence alignments, and that we could compute efficiently directly on the multiple sequence alignment. So we introduced another order which is based on the most divergent columns of the multiple sequence alignment. So the idea is that you select the n most divergent columns of the multiple sequence alignment, and then you will use the nucleotides in those columns in order to reorder your sequences. So to take my usual example, I spot the 3 most divergent columns in my multiple sequence alignment. So this one is the most divergent with this one. And the third most divergent is this one. So I will take the nucleotide here in this one, U, A, and G. So I extract a subword from each genome like this. And then I will reorder depending on those subwords. So we call them discriminative subwords of length 3 in that case. And we reorder depending on those subwords, so like this. And now I have a multiple sequence alignment where the redundancy somehow have been clustered together. And we have a nice a multiple sequence alignment, which is as good as the optimal one in that case. Of course, not in the general case, in this example. So we see how it can improve somehow the compression after that, because we will store this more efficiently. So we've used a data set of 1.9 million SARS-CoV-2 sequences. So we started from the Hunt et al. Data set of 4.5 million, and we removed quite extensively some sequences in order to have a multiple sequence alignment without too many gaps. We aligned them using HLine 3, and we obtain a multiple sequence alignment of about 35,000 columns, while the genome is of about 30,000 base pair. So this corresponds to a multiple sequence alignment of 65 billion characters, so it's quite a large multiple sequence alignment. And if you want to consider all the possible pairs of columns in your multiple sequence alignment, you have to consider 607 million pairs of columns, knowing that each column is of 1.9 million letters. So that's quite a huge amount of data to consider. Regarding time and space efficiency for construction, we've compared our approach, CREMSA, with 2 generic compressors, gzip and xz. And we've also compared to a specific compressor dedicated to multiple sequence alignment, which is called CREMSA. And we've been able to build our structure in smaller amounts of time compared to the compressors, using memory that is larger than gzip and xz, but much smaller than CREMSA, and which is very reasonable. You can run it even on a Raspberry, I guess. I've not tried. So that for construction. And regarding space efficiency, so you have the compression ratio, so the higher the better. gzip is not good for reasons I could explain. xz and CREMSA are quite similar in terms of compression ratio, and CREMSA, which is a specific compressor, is better than the alternatives. But we can also see what happens when we change the order of the sequences. So using the phylogenetic order, it improves for xz and CREMSA about the same margin, and it also improves slightly for CREMSA. And then our third order, the discriminative order we introduce, it doesn't change much for xz, so it improves compared to the initial one, but it's not better than the phylogenetic. But for our structure, it also improves compared to the phylogenetic order, so it's quite a nice property that we've been able to introduce an order that is better for our structure than the phylogenetic order. And in the end, we are not that far from CREMSA. If you want to know the size of the structure itself, so for the 65 billion characters multiple sequence alignment, we store that in 24 megabytes, while CREMSA is at 13 megabytes. So we are at about 12 bytes per genome, which is not much. But we're not just compressing data. We also want to query our multiple sequence alignment, and so that's what we do. And so we cannot compare to the others because the others are compressors. So we can do a random access to our multiple sequence alignment at a rate of about 2 to 3 megabytes, depending on the order of the sequences. But what we are interested in is computing number of nucleotides in pairs of columns. So that's what is useful to compute covariations. And as you can see, we are able to do that in about 100 microseconds or even less for a single pair of column. And you can see also the improvement with the order. We have a 5-fold improvement between the initial order of the multiple sequence alignment and our discriminative order. So this shows that it's really improves both in time and space. It was not just a theoretical property. So in the end, we have been able to compute our multiple sequence, our correlations for all the possible pairs of columns for the SARS-CoV-2. So it took about 10 hours on a single thread. And we show here 2 different correlation scores. So again, I won't enter into the detail because it's not the scope of the talk, and there is also still some work remaining on this part of correlation scores. But here what we show is the correlation score above a given threshold between any pair of columns in the multiple sequence alignment. And for the readability, we've used buckets of 1,000 nucleotides. So you see that there is a spot here towards the end where there are a lot of possible correlations, and it's where you have the spike gene of the SARS-CoV-2 that is quite well known as a gene. So in the end, we have a data structure that both compresses and provides efficient queries on multiple sequence alignments. So I've shown here only data on SARS-CoV-2, but we've also applied it to HIV and proteins. So we are not restricted to SARS-CoV-2, even if it makes more sense on SARS-CoV-2 because you have a huge amount of data. As we've shown, reordering saves both time and space, which is quite a nice property. And we've introduced a new order, the discriminative order, in order to improve both time and space. And I think there is some work to do regarding this order because we had some surprising results with this order. We can discuss later if you want. And we've been able to compute correlation scores on the pairs of columns. We've done pairs of columns yet, but actually we can extend to any numbers of columns. And I mean, it would make sense not to consider just pairs of columns, but more columns in order to have a more robust signal, because among in the figure I've shown you with all the correlations, I mean, some of them are probably, and maybe most of them are probably false positives. But if we take into consideration more columns, we would have a more robust signal, and we could do that with CREMSA, with now that we have an index built on the multiple sequence alignments. This is something quite easy to do. And this is available on GitLab if you want to use it with Docker images. Thank you.
Thank you. So we have time for a few questions. If you are interested to pose a question, go to the microphone. So we will have the first question at microphone 2 here.
Thank you for the very nice talk. I was very surprised with the result that comparing phylogenetic compression with subwords. Could you explain why it works better with subwords?
Yeah. So yeah, you're mentioning this point which is above the phylogenetic order, yes. Right? Okay. Yeah. So our order is better, I think, for technical reasons, because the phylogenetic order we could think, okay, it should be the perfect or near perfect order, but the genome we get are not perfect sequences from the start to the end. Some of them are slightly shorter than the 29,000.9 base pair. So you have gaps at the ends of the genomes, and this is an information that is not taken into account with the phylogenetic order, obviously. But our measure can take that into account. And so I guess this helps to have, for instance, gaps that are well aligned in the multiple sequence alignments. And so we can improve over phylogenetic information thanks to that. So to state it quickly, I think we can take into account technical artifacts that a phylogenetic measure cannot.
Thank you.
Sure. Thank you for the very nice talk. So I first have a follow-up on this question. So what if you change the number of 5,000? What if you just have only 500 or 1,000 or even more? What would it matter?
I have a plot for that.
Thank you.
Here is the answer. So you have the discriminative order. So the 5,000 is about around here. So that's the 5,000. So you can do less. And this also gives me the opportunity to mention that random order is no, not random order. Choosing columns randomly is worse, obviously. And what is surprising in the discriminative order is that if you don't take the columns in the order of the divergence, but if you shuffle them, if you take them in the shuffle order, so you take still the most divergent columns, but in shuffled order, you can get a compression ratio that is higher than taking them in order. We don't have an explanation yet, but that's what we observe, and we observe it in several data sets, not just on this one.
Thank you. The question I have is like, this kind of reduces the pairwise quadratic complexity to kind of linear, but afterwards you are using RNA alifold, which is still a cubic time algorithm based on the length of n, right? So it's still n cubic to fold it in RNA alifold. So I'm suggesting maybe you should use a linear time version like ling alifold or linear alifold, which scales linearly with the sequence length after your compression.
I didn't get what you mean. On what aspect do you suggest a linear algorithm?
So your RNA alifold runs in a time that scales cubically with the sequence length. So it's n cubic, where n is like 30,000 for SARS-CoV-2.
We didn't run RNA alifold here. We just
the last figure.
It was just the conservation scores.
Yeah, right. But like your motivation is like so that you can run RNA alifold maybe based on your compression.
Not necessarily. It's already to spot areas of the genomes where there may be interesting stem loops.
I see.
And we could run RNA alifold, but just on smaller areas, not on the whole genome, I guess.
Right. I see. So you're just interested in the covariation column. Thank you very much.
Sorry, we are running a little bit out of time. So if there is a shorter question, maybe.
Right. Thank you for the talk. Quick question. I wanted to know the reason why you filter unknown nucleotides, right? Is it a computational reason or is it just quality of the data?
Okay, there may be a computational reason because we needed 1.1 terabytes of main memory to run the multiple sequence alignment, and so we did not have a server with so many terabytes of main memory, so we could not have coped with a larger number. The other reason is that if we take all the sequences, the MSA is crap. I mean, you have gaps everywhere, so you need still to filter. Maybe we could have filtered less aggressively, but yeah, we need to filter the sequences.
Okay, so maybe Nicola can come to get ready. And I have one more online question, which says that if a column has more than 2 states, so the 10 transitions are not the same, how the original sequences could be retrieved? So this is probably some clarification question from online audience.
About retrieving the original sequences?
So
right?
How can you retrieve original sequences if you have more than 2 states in a column?
Well, we have random access to any nucleotide, so you can recover a sequence by traversing all the lines. So yeah, we can do that.
Okay, so let's thank the speaker again.
Thank you.